Let's look at the myocardium. There are two types of cardiac muscle cells, or cardiomyocytes. These are contractile and non-contractile cells. The difference between these two is that the contractile cells are involved in contraction and allow the heart to function as a pump. The non-contractile cells are not involved in contraction. What they do is they either initiate an action potential or transmit that action potential along an intrinsic conduction system. We'll talk about this in a minute. Let's look at the microanatomy of cardiomyocytes. We'll begin with some general characteristics. Remember that cardiac muscle has intercalated discs. These are located between adjacent cardiac muscle cells. These intercalated discs consist of two types of membrane junctions. Gap junctions, which are like little tunnels between the two adjacent cells. These gap junctions are made from proteins that are part of the cell membranes of the two adjacent cells, and this allows ions to travel from one cell to the other without having to go across a cell membrane. Gap junctions and the fact that cardiac muscle cells are branched allow the two atria to contract as one unit. This is a syncytium, and the two ventricles to contract as one unit as a separate syncytium. The second type of membrane junction found in the intercalated disc are called desmosomes. Intermediate filaments of adjacent cells are basically sewn together. These are anchoring junctions, and they prevent the cells from being pulled apart when the cells are contracting. A syncytium means that all of the cells are going to contract at the same time. So as mentioned before, the gap junctions allow the two atria to contract at the same time as a single unit. So you have an atrial syncytium, and then the two ventricles will contract at the same time as a unit. So you'll have a ventricular syncytium. And this is due again to the fact that the cardiac muscle cells are branched, which allows the action potential to travel from one cell to multiple cells. And the presence of the gap junctions allows the action potentials to travel very quickly. The atria are going to contract before the ventricles contract, and the atria are going to function as primer pumps. I'll explain to you later what this means, but basically, when the atria contract, they push additional blood into the ventricles, and this will stretch the walls of the ventricles. And this increases the strength of the contraction of the ventricles. But we'll come back and look at that a little bit later. But they are primer pumps, the atria are. Recall that the mitochondria are the powerhouse of the cell, and this is where aerobic cellular respiration occurs. This is how most of the ATP is synthesized throughout your body in aerobic cellular respiration. The heart synthesizes the majority of its ATP almost exclusively by aerobic cellular respiration. So over 95% of the ATP that is made by the cardiac muscle cells will be done aerobically. For this reason, you're going to have a lot more mitochondria and cardiac muscle cells compared to skeletal muscle cells. So let's look at the difference. Let's compare the amount of mitochondria by volume. So about 25 to 35% of the volume of a cardiac muscle cell consists of mitochondria compared to 2%. The primary energy source used for aerobic cellular respiration in cardiomyocytes is fatty acids. This is due to the fact that cardiomyocytes do not store glycogen, which is the storage form of glucose. The high volume of mitochondria within the cardiomyocytes allows the cells to synthesize a lot of ATP. This gives them resistance to fatigue. This is important since the heart has to pump continuously throughout your entire life. Let's look at the non-contractile cells. Pacemaker cells exhibit autorhythmicity. They are located within the intrinsic conduction system. And what this autorhythmicity means is that auto means self. So these cells, the pacemaker cells, have the ability to initiate an action potential without neural input. And this is different from skeletal muscle cells. Remember that skeletal muscle cells have to be innervated and stimulated by motor neurons. These pacemaker cells can generate their own action potential. They don't need help to do that, and that's the auto part. There is a rhythm. In other words, they depolarize so many times per minute, and this is the rhythmicity. So pacemaker cells have autorhythmicity, and read about this in your textbook, be able to tell me about it. And also, after we go through it, you should be able to tell me why this is possible. Let's review action potentials before we look at the action potentials within pacemaker cells. Hopefully this looks familiar to you. This is an action potential that you would see in a neuron. Now let's review. A neuron that is not being stimulated has a resting membrane potential. This membrane potential is polarized. The inside of the membrane is negative compared to the outside. This is due to a higher concentration of positive cations in the extracellular fluid and negative proteins inside the cell. A stimulus will open up voltage-gated sodium channels, allowing positive sodium ions to move inward. This begins to depolarize the membrane. When threshold is reached, an action potential begins. The first phase of an action potential is depolarization. The inside of the membrane is beginning to become more positive, less negative. Let's move from depolarization to repolarization. During repolarization, remember that the voltage-gated sodium channels close and the potassium voltage-gated channels open up. Potassium is on the inside, so now it begins to leave the cell. There's an efflux of potassium, and as it leaves, it carries with it its positive charge. This makes the inside negative once again, so you are repolarizing the membrane. However, at this point, you have potassium primarily on the outside and sodium primarily on the inside, and this is opposite from the resting membrane potential. In the resting membrane potential, recall that the sodium is primarily on the outside and the potassium on the inside. So now we're going to have to put everything back in the right place, and this is going to involve the sodium-potassium pump. We have to return the membrane back to the resting membrane potential, or return it back to being polarized. The pump is going to pump 3 sodiums out while it pumps 2 potassiums in. Let's look at the pacemaker cells. In the pacemaker cells, you begin with what's called a pacemaker potential. There is no resting membrane potential. It is unstable. So in the pacemaker potential, you have sodium channels that are open, and they open in response to hyperpolarization. Recall that hyperpolarization is when the membrane potential becomes more negative than at rest. So in response to hyperpolarization, which is due to the potassium ions moving out, you're going to have a pacemaker potential. At this point, the potassium channels are closed once you begin the pacemaker potential. So what you're going to have to be able to do is tell me which channels are open, which channels are closed, and be able to identify the phase here in an illustration. So if I show you a graph, be able to label it for me. And tell me in which direction the ions move. Once threshold is reached, so the sodium as it moves inward is making the membrane potential less negative, and once threshold is reached, which is around minus 40 millivolts, a second set of channels open up, and these are calcium voltage-gated channels, and this leads to the rapid depolarization of the action potential. So remember, you have to reach threshold before you have an action potential. So notice that everything above threshold is an action potential, and it is the influx of calcium that leads to the depolarization of the action potential. So if I ask you what ion is responsible for the rapid depolarization of the pacemaker cells, I want you to be able to tell me it is calcium. And calcium moves inward, so you have an influx of calcium. Notice at the top of the graph, the calcium channels close and the potassium channels begin to open. This leads to repolarization. And so now you're going to have an efflux of potassium. So potassium is leaving the cell, returning the membrane potential back to negative, making it more negative. And then we're going to have the hyperpolarization. So if you notice down at the very bottom there, the hyperpolarization, which then will open up the sodium channels, and you begin with the pacemaker potential. So this is a continuous cycle. So there is a pacemaker potential at the end of the red arrow. That is going to be the rapid depolarization that leads to the action potential. And that, recall, is in response to a calcium influx. And this is going to be the repolarization that is due to a potassium efflux. Now let's look at the heart conduction system. The heart conduction system is called an intrinsic conduction system. Remember that the term intrinsic means that it's within the organ. This system consists of cells that initiate action potentials and transmit or conduct the action potential. Once the action potential is transmitted along the conduction system, it is also then dispersed to the rest of the contractile cells. So the intrinsic conduction system is going to consist of non-contractile cells. Let's take a look. The sinoatrial node is going to be located adjacent to where the superior vena cava empties into the right atrium. When it fires, this node consists of pacemaker cells. And when they fire, the action potential is transmitted to the next step along the intrinsic conduction system, as well as over to the left atrium. So throughout the right atrium and then over to the left atrium. And remember that the action potential can travel very quickly because you have cells that are branched and you also have the gap junctions at the intercalated disc. So both of the atria will contract as a syncytium, the atrial syncytium. As the action potential is traveling from the right atrium over to the left atrium, it is also traveling to the next step, as I mentioned before, in the conduction system. And this is going to be called the atrioventricular node. But let's look at the SA node in greater detail. The SA node is called your pacemaker of your heart. And the reason why it's called the pacemaker of the heart is because it has the fastest depolarization rate of all of the pacemaker cells found within the conduction system. It's going to have a depolarization rate, in other words, a beats per minute of between 80 to 100. However, you know that your resting heart rate is usually lower than 80 beats per minute. So the intrinsic heart rate or the intrinsic depolarization rate of the SA node is going to be between 80 to 100 beats per minute, but the autonomic nervous system can influence it. We're going to see a little bit later how the parasympathetic nervous system will decrease your heart rate, and this is what it's in control most of the time, and the sympathetic nervous system will increase your heart rate. But nonetheless, the SA node is the pacemaker of the heart because it has the fastest depolarization rate of between 80 to 100 beats per minute. From the SA node, as mentioned before, if we continue down the conduction system, the action potential then will go to the atrioventricular node, which is going to be located on the posterior wall of the right atrium, adjacent to or close to the right atrioventricular or tricuspid valve. The AV node is going to have a depolarization rate of between 40 to 60 beats per minute. So 40 to 60 beats per minute, but there is a delay here. It's a 1/10 of a second delay. This delay is critical. This delay allows the true atria to contract as a syncytium, filling the ventricles prior to their contraction. From the AV node, the conduction pathway then moves into the interventricular septum. And the interventricular septum is the wall between the two ventricles. to what's called an atrioventricular bundle, or the bundle of His. This has a depolarization rate of about 30 beats per minute. It's not fast enough to be efficient. And then from there, you will have a branch, a right and a left bundle branch, and this continues down through the interventricular septum up to or down to the apex of the heart. And then notice it starts moving back up the myocardium of the ventricles into what is called the subendocardial conducting network, which is made up of very large non-contractile cells called Purkinje fibers. And this transmits the action potential to the contractile cells of the ventricles. The ventricles then will contract as a syncytium. They begin contracting at the apex and move upward. So keep in mind that the non-contractile cells are part of the intrinsic conduction system. Let's do a little review. What are the two types of non-contractile cells? I mentioned the first, and those are the pacemaker cells. The second type of non-contractile cells are just called conducting cells, because their job is to conduct action potentials, just to transmit those action potentials. So you have pacemaker cells and conducting cells. Let's look at some imbalances. I'm going to ask you to stop the video and write down these imbalances and then look up their causes and be prepared to talk about them. So I want you to look up fibrillation, ectopic focus or foci, heart block, tachycardia, and bradycardia. So at this point, pause the video, write down these terms, and look them up. Let's look at action potentials within contractile cells. I have a skeletal muscle on the left and a cardiac contractile muscle on the right. So let's start with the skeletal muscle cell. An action potential in the skeletal muscle cell involves depolarization and repolarization. So depolarization, number one, is due to sodium influx. So the sodium voltage gated channels are open at this point. And repolarization is due to voltage gated potassium channels being open and potassium leaving or exiting the cell. And this is an efflux of potassium. You can see here resting membrane potential is minus 85 millivolts. And actually this one shows you hyperpolarization. Notice how on the right side, when you're repolarizing the membrane becomes slightly more negative than the minus 85. That would be a hyperpolarization in the skeletal muscle fiber. And then it returns back to resting membrane potential. Now let's look at cardiac muscle cells. I'm going to go through this in greater detail, but just in general. Phase one is depolarization. At this point, both sodium and calcium channels are opening up. However, these are slow calcium channels, so they actually don't completely open up until we get to number two. So depolarization is due to an influx in sodium. Then you're going to have a plateau phase, and that is where you have a leveling off there. This plateau phase is unique. It's different from skeletal muscle fibers, and we're going to talk about the importance of that. Notice in the plateau phase, I have a section with two dashes there. What this is telling me is that this plateau phase extends, so they've just removed part of the plateau phase just so that they can fit it on a small graph. So the plateau phase is due to calcium influx. I'll talk about the fact that there are a few potassium channels that are open here, and this is causing just a very brief repolarization. I'll point that out in the next slide. And this leads to a plateau phase where you have a leveling off of the membrane potential, where it remains depolarized for an extended period of time. And then phase three is the repolarization phase. Calcium channels are closed at this point. The rest of the potassium channels open up, and you have the exit of potassium, which is going to bring about the repolarization. And you're going to return it back to resting membrane potential. Now this is going to be different than pacemaker potentials because in pacemaker potentials you don't have a stable resting membrane potential, but here you do. So let's go through the action potential and the contractile cardiomyocytes step by step. So the first phase is depolarization, as I mentioned before. And during this phase, you're going to have both sodium and calcium channels opened. However, remember the calcium channels are slow calcium channels, so they don't completely open until we get to the plateau phase. So at this point, depolarization is going to be due to the influx of sodium. Phase 2 will be the plateau phase. There is that brief period that I've highlighted in yellow that is a slow repolarization, and this is due to the fact that a few of the potassium channels are open, and so you have some potassium that is leaving the cell. However, you have more calcium that is moving into the cell because at this point, the calcium channels are completely open and you have an influx of calcium. So calcium is moving in, and this is what's responsible for the plateau phase. But that very brief repolarization highlighted in yellow is due to a small efflux of potassium. And then at the end of the plateau phase, the calcium channels close and the rest of the potassium channels open up, and that leads to repolarization. And this is due to the efflux or the exit of potassium. So the membrane is moving toward becoming more negative, and then you're going to return it back to the resting membrane potential. So why is that plateau phase important? That plateau phase will extend the period of the action potential. So we're going to have a longer action potential in cardiac muscle cells compared to skeletal muscle cells. In skeletal muscle cells, the action potential may last around 2 milliseconds, whereas in cardiac muscle cells, it can last 200 to 500 milliseconds. During this period, you cannot have another action potential. In other words, regardless of whether you're trying to stimulate the cardiac muscle cells or not, they cannot respond to the stimuli. And so this is going to create a long absolute refractory period, which prevents tectonic contractions. Remember in skeletal muscle cells that as you increase the frequency of the stimuli, you can get to a point where you have a sustained contraction, and this is called tetany or tectonic contractions. You can't do this with the heart because this would kill you. So you're going to have this long refractory period, and this is due to the plateau phase. Be sure you can tell me why the plateau phase is important. Let's look at excitation-contraction coupling in cardiac muscle cells. Excitation-contraction coupling in cardiac muscle cells is very similar to what happens in skeletal muscle cells. I'm going to encourage you to go back and look at this in your textbook just as a review. Let's talk about what I have here. Down at the bottom in this slide, you have an illustration of a sarcomere. Remember, the sarcomeres are the contractile units located within skeletal muscle cells. Cardiac muscle cells have the same thing. And this is what makes these cells striated. The blue lines represent the C disc, and these are the boundaries of a sarcomere. The red lines here represent the thin filaments, and I'll come back to those in just a minute. And then in the center, the gold and green lines represent the thick filament, which consists of myosin proteins. And these are double-headed. So let's look at the thin filament. Up at the top, I have an illustration of the thin filament. The main protein in the thin filament is actin. This is a globular protein arranged in two strings that are twisted, and these look like a double strand of pearls that is twisted. Each one of those actin molecules is going to have a binding site for the myosin head. So remember, those are the green structures on the thick filament. Troponin is another protein, and you can see that we have three subunits here, C, I, and T. Troponin has a binding site or has binding sites for calcium. And associated with the troponin is tropomyosin. This is this long filamentous protein. And what the tropomyosin does is it covers up the binding sites on actin. So as long as the tropomyosin is covering up those binding sites on the actin, the myosin heads cannot bind. And those myosin heads have to be able to bind in order for a contraction to occur. So what happens here? When calcium binds to the troponin, this changes the shape of the troponin, moving the tropomyosin away from the blocking position. And now the binding sites on the actin for the myosin heads are exposed. The myosin heads will bind, and we're going to then have contraction. So as long as calcium is present, you're going to be able to have a contraction. Notice that you have the I and the T troponin units or subunits. Whenever you have a patient that has had a myocardial infarction or a heart attack, you can order blood work to see whether you have troponin I and T in the blood. If these enzymes are present in the blood, then that means the individual has had a myocardial infarction, because damage to the cardiac muscle cells will cause the release of these enzymes. So that's important for you to know where the troponin is coming from. Let's just quickly review excitation-contraction coupling in skeletal muscles using this illustration. So you'll notice over on the left, you have a motor neuron with the axon terminal. And in the axon terminal, you're going to have synaptic vesicles that are storing the neurotransmitter acetylcholine. The sarcolemma is the plasma membrane of the skeletal muscle fiber or cell. And notice the place right underneath the axon terminal. This is called the motor end plate. And there is space in between the motor neuron, the axon terminal, the motor neuron, and the motor end plate of the skeletal muscle fiber. And that's the synaptic cleft. So what happens here? Remember that an action potential stimulates the release of acetylcholine from the axon terminal. Acetylcholine diffuses across the cleft and binds to receptors on the motor end plate. And this is going to cause a motor end plate potential, and the sarcolemma or the plasma membrane will be depolarized. If it's depolarized to threshold, you're going to have an action potential. So this is what we have here. The action potential travels along the sarcolemma, down T-tubules, and these are the green structures. And associated with the T-tubules, you're going to have the endoplasmic reticulum, and in skeletal muscles, that's called the sarcoplasmic reticulum. And the sarcoplasmic reticulum stores calcium ions, and you can see those there. So as the action potential is traveling down the T-tubules, this stimulates the release of the calcium ions into the cytoplasm of the cell. So this is where the calcium is coming from that is going to bind to the troponin. An action potential is required in order for the calcium to be released from the SR. Once the calcium binds to the troponin, you're going to then have the movement of the tropomyosin away from the blocking position. The myosin head here in green will be able to bind, and we're going to have what's called a power stroke. And remember, this is going to slide the thin filaments inward, and this is the contraction. Go back and review this in your book if you don't remember this from AP1. Let's look at what happens in cardiac contractile cells. So it's going to be a little bit different. And what I'll want you to be able to tell me is, what is the difference here? So you see the sarcolemma here. Everything up to this point is the same. We're going to have an action potential that's been generated along the sarcolemma. It's going to go down through the T-tubules. In cardiac muscle cells, you don't have as extensive of a T-tubule system as well as the SR. What is different here is when the action potential travels down the T-tubule, it causes the opening of calcium channels. These are voltage-gated channels, and calcium moves from the from the extracellular fluid, which is the blue part here. So from the extracellular fluid through the calcium channels into the cytoplasm. The calcium that moves inward actually is what causes the release of calcium from the SR. So this is the first difference here. It is the movement of the calcium inward. That calcium is necessary in order to cause the release of calcium from the SR. About 10 to 20% of the calcium used during the excitation-contraction coupling and what is called the sliding filament mechanism, in other words, the shortening of the sarcomere, is going to come from the extracellular fluid. So 10 to 20% of the calcium required comes from the extracellular fluid. The remaining 80 to 90% is coming from the SR. So not all of the calcium comes from the SR. You need calcium in the extracellular fluid. It is critical that you understand this because we're going to be looking at how extracellular calcium levels can influence the heart, the contraction of the heart. Now, once the calcium leaves the SR and you have the calcium from the extracellular fluid, that calcium will do exactly the same thing as in skeletal muscle. It will bind to the troponin, and this will cause the tropomyosin to move away from the blocking position, allowing the myosin heads to bind to the actin. The last slide for this module is on the electrocardiogram, the EKG or ECG. Electro refers to the fact that this is a reading of the electrical activity of the heart. Here is a graph of an EKG. For the test, I will want you to be able to label this graph and tell me what is occurring at each point in the graph. Other than that, I'm not going to ask you to tell me anything about imbalances. This is a normal EKG. So let's look at the different activities in this graph. We're going to begin with the P wave. That's the green wave here. The electrical activity in the heart occurring during the P wave is atrial depolarization. The QRS complex is the reading during ventricular depolarization. And the T wave, the blue line, is the reading during ventricular repolarization. Notice that there is no atrial repolarization. That's because it is occurring during the QRS complex, and the QRS complex is hiding it, so you can't see it. So you're not going to be able to see atrial repolarization. It is not on the graph. This concludes the first module for Chapter 18.